Hi, I'm Ben Joris from the GLBIO lab at the University of Western Ontario, and this is my lightning talk on my research into bacterial conjugation in the human gut microbiome. Type 4 conjugative elements are genetic elements that can be exchanged between bacterial cells and are widely known to carry detrimental metabolic pathways such as antimicrobial resistance or virulence factors. As such, it makes precise identification and quantification essential when looking to compare human gut microbiomes between cohorts. I sought to develop and employ methods for identifying conjugative systems in a context-independent manner. The first method was employed to 101 de novo assembled metagenomes from two cohorts, a general North American and a North American preterm infant cohort. In this method, I used profile hidden Markov models for conjugative proteins and identified genetic fragments with matches, and then quantified these matches. I also annotated a set of 2,500 bacterial genomes found in the human gut and searched for genetic fragments containing annotations for conjugative systems. I then quantified the relative abundances of these conjugative systems across 8 different cohorts. From the de novo assembled metagenomes, the abundances of the assembled conjugative systems are starkly different between the two cohorts, with the systems appearing to be almost mutually exclusive between the two groups. Now, this is not wholly unexpected because the preterm infants are on a steady dose of antibiotics from birth, killing the natural gut flora. In addition, infants generally have a different array of commensal bacteria than adults. As you can see on this circular phylogram, the preterm infants have a high signal in the proteobacterial conjugative systems that likely originate from hospital-acquired antibiotic-resistant pathogens, whereas the general cohort doesn't appear to have any of these systems, only having those belonging to the common commensal phyla. I aligned reads from 785 metagenomic samples from 8 cohorts to the conjugative systems I identified by annotating a genome set of 2,500 bacterial genomes. The two cohorts that stand out the most from the rest are the North American preterm infant cohort, which is the same as the previous figures, and the European infant cohort. Both of these cohorts have virtually no reads mapping to any of the identified bacterial conjugative systems. The West African and South American cohorts also stand out from the other non-infant cohorts, showing similar patterns of conjugative systems to one another that are distinct from the other non-infant cohorts. The remaining 4 cohorts appear to be roughly equivalent on this plot. I additionally wanted to see how often the conjugative systems I identified are included in metagenomic assembled genomes because it is commonly employed metagenomic analysis for evaluating the metabolic potential of a microbiome. As you can see from this flow diagram, only about a quarter of the identified systems are included in these metagenomic bins, which are an approximation of bacterial genomes. In contrast, the background binning rate of non-conjugative sequences that are greater than 5 kilobases in length is about 70%. This means that conjugative systems are being systematically excluded from the binning process. From my research, I have found that conjugative systems are distinct between population-based cohorts and can be used to cluster and separate groups. Additionally, the stark differences between adult and infant cohorts points to the potential as a developmental biomarker. Because conjugative systems are included at a lower rate than non-conjugative contigs in metagenomic bins, special methods must be used prior to binning in metagenomic analyses to ensure that they are not missed. Beyond the geographic focused cohorts that I examined, bacterial conjugative systems could be the explanatory difference for microbiome-linked human health outcomes, and if the researcher is using a mag-focused approach, they could come to the incorrect conclusions from their data. Thank you for listening to my presentation. Please feel free to leave any question. I would be more than happy to answer them for you.